Welcome back. In this video, we're going to be working through the process of science. So this is the second section of chapter 1. We're going to consider the range of the natural sciences. Then we'll look specifically at how scientific inquiry is done while we consider the types of reasoning needed to do it. And finally, we'll get to look at the differences between basic foundational science and applied science, and then how we as scientists then get that information out to other people, other scientists and general audiences. All right, so the discoveries of biology are made by a community of researchers. This idea of the lone scientist making discoveries in their lab is fiction. Science isn't done that way. Science almost has never been done that way. Even way back in history, we tend to hear these big names of scientists, but they had teams of people they were working with usually. So those teams start at the lowest of levels, right? You have a research lab at a college, university, and you have a whole team of people in there. You have a lead researcher and then the people that work under them. And then it's also still a community as we move up from there, from say the department of a university, say the biology department. All those people work together and help each other, even if their research is different. And then when we publish our work, we're sharing it out to the even larger scientific community. People all over the world can see the work that we've been doing. This is a really important part of science, this sharing. We put it out there for our colleagues to review, to see if there are any problems with the work we've done, to make sure that we've done it well, that it's meaningful. And then to be able to take the work that I've done and for someone else to get an idea from it and take the next step. That's incredibly important, and a lot of people don't think about that part of it, but we share our knowledge so that we can continue to build more knowledge. The sciences, and those of us who engage in it, we're often seen as just knowledge machines. I hear that from people a lot. Oh, you just know so much stuff. We're just churning out facts. It's like, well, yes, you have to have a large foundation of knowledge to do this work, but that's not what the science is. That's the foundation of the science. The reality is that research science is an incredibly imaginative and creative field to be in. You have to develop good experiments that can be replicated by other people to discover new things. It's a really intricate process that involves a lot of failure, in a way that a lot of other fields don't. You spend a lot of time not getting an answer, and you have to keep trying and persevering, push through until you figure out a way to get an answer to the question you're working with. Now, some of the sciences focus on solving real-world problems. We call this applied science, and it's probably the science you're most familiar with. Usually we think about studying diseases and how to cure them. But a lot of science is just based in curiosity. How does this work? Why did this happen? And that kind of science actually has a huge impact on the applied sciences. We'll accidentally discover things, and it will have this huge, long-ranging impact on all sorts of other things. We'll look at an example in just a little bit. So we don't often take that moment to define things like science, because even for the non-scientist, it feels like a familiar term. I know what science is. I know what biology is. But let's do some clear definitions here. So science is knowledge about the natural world. And biology is a subset of the natural sciences that focuses on the living parts of the natural world. It's also important that we know what science isn't and what it can't do. So we don't use the process of science to answer purely moral or aesthetic or spiritual questions. And the reason is the context. So depending on where you stand, there are a lot of different answers to moral questions and aesthetic questions, spiritual questions. So we can't really test them or observe a phenomenon related to them. For example, let's say that I put up a painting and, this is my favorite painting, I just love this. Well, some of you aren't going to like it, right? We can't apply the scientific method to that process. Now, sure, we could do a little bit of light polling. Oh, lots of people like this, but that's not necessarily scientific. okay? And that sometimes really gets people. That's opinion, and we might be able to say, well, the majority of people have this opinion or believe this thing. That doesn't make it scientifically valid. So it's just an important distinction for us to make. In the sciences, we're looking for answers. We're looking for answers to how something worked or why something happened the way it did. To do this work, we have to be able to observe the phenomenon. We've got to be able to test it. We've got to be able to measure it. So in order to do that, it has to be an observable physical phenomenon. And moral and spiritual things, that's really more, that's the realm of philosophy, and we'll leave it to them. So in order to do scientific testing, it helps if we have a clearly defined method, and we do. The scientific method, you're probably familiar with it. I'm going to give us a quick summary. We'll talk about it in more detail a little bit later. Hopefully this is more of just a refresher from high school. So scientists, we tend to use the same general steps to find answers to our questions. We call it the scientific method. So in the scientific method, we're going to have these defined steps that involve experiments, careful observations. Now, it's kind of a guideline because this process is very fluid. We might develop a hypothesis and test it, and the results may not come out in a way that we can use them the way we thought we would be able to. So we would have to do a new experiment, new hypothesis, and this process goes on and on, feeding into itself. So it's not a one and done kind of thing. It tends to be very elaborate and complex. A hypothesis, hopefully you remember, is just a suggested explanation for an event that can be tested. We have to be able to have a yes or no answer at the end of it. If no isn't possible, then it's not a hypothesis. We'll get into it more. So scientific theory, which this one gets people, this is a generally accepted, thoroughly tested, and confirmed explanation for a set of observations or phenomenon. All right. Things like cell theory, that the basis of life is cells. It's not a guess. All right. Like that's, sometimes people use the word theory like, well, in theory, I could jump 10 feet without a trampoline. Like, oh, okay. A guess, a thought, something that's kind of just off the cuff. In the sciences, a scientific theory is well supported, well documented, well known. Most of the big scientific theories are 100 years old or more, some of them. The theory of gravity, like we don't question the ones that seem very easily observed to us, but there are lots of other ones, and we'll, not so much in this class, but if you take the second half, we'll talk a lot more about evolution. The theory of evolution is well established, well accepted, and yet we still have people say, oh, scientists don't really know. No, we do. It's well established. So we'll talk about some theories. We tend to use that word theory more in the life sciences. We use the word law a bit more frequently when we're talking about physics, chemistry, math. But they're really more or less the same thing. Scientific laws are going to describe how elements of nature will behave under certain specific conditions. So usually a law is going to have some sort of mathematical formula you can assign to it. We'll talk about some of the laws of thermodynamics later, and they have clearly defined formulas that you can work with. But laws and theories both function under the same kind of umbrella of well supported, well documented, widely accepted. All right. So there are a lot of different scientific fields that fall under the natural sciences umbrella. Astronomy, biology, chemistry, computer science, geology, physics, mathematics. Those are kind of the big ones, right? The broad terms for fields. At the higher levels, we're going to divide these down into even more specific subfields. That didn't used to be the case. A couple hundred years ago, if you were a biologist, you were an expert in all things biology. But what we knew and understood about biology was a lot smaller. Same with chemistry and physics. Just our breadth of knowledge today, even just the last 50 to 80 years, is just huge exponential growth in our knowledge base. So if you're a biologist like me, I have a very solid foundation in all of biology at the lower level, right? So I'm here teaching you about biology because I'm an expert in biology. But my higher level, my more specialized knowledge is in cell biology, okay, the study of individual cells. And then that actually goes even a step further. My ultimate specialization is in cell motility, so how cells move, both environmental, like free living cells out in ponds and lakes and stuff, and also the cells like inside your body, like how your immune system cells work. Okay, so that's quite specific, how cells move. But what's really interesting is, in order to really understand how cells move, I also had to have a really strong educational background in physics and chemistry, especially the chemistry, in order to do the experiments and to study how the cells were moving, how they were generating their movement. You got to know the chemistry. So not only do scientists have to specialize in their own field, but sometimes we have to do more specialization in other fields too. So hopefully that helps you better grasp how long it takes to become a scientist. That all that knowledge, it's not just textbook knowledge. Of course, there is lots of textbook knowledge you have to acquire, and then lots of research papers, like published papers, that you have to be very familiar with. But it's the practical at the bench work that really is what elevates you as a scientist, what allows you to take all that knowledge you've gained and apply it directly to creating experiments and producing results. In order to become an independent scientific researcher, you're going to have 10 or more years of advanced level training. It's not something that you can just sometimes you get armchair scientist. You can't just read textbooks and watch videos on YouTube and become a scientist. You can have a strong interest in science and enjoy science doing that, but to become a scientist requires more than that, more than just the knowledge. You need the practical experience as well. So it's usually, like I said, at least 10 years of advanced level training, and even more before you might have your own research group that you run and design what kinds of experiments and things are going to be done, and you're training other people how to become scientists as well. All right, so I said it's not just textbooks, right, that we're doing. So what are scientists doing? Well, part of the training involves learning about reasoning and logic. So inductive and deductive reasoning are two kind of foundations. So in inductive reasoning, it's a form of logical thinking that uses related observations to arrive at a general conclusion. All right, we look at what's already there, and then we develop conclusions based on current evidence. This is how a lot of the research on how the human brain works. That's how it's done. They do fMRI of hundreds of different brains all performing the same task, and then look for the commonalities, right? So this is typically considered descriptive or discovery science. And then we have deductive reasoning. So this is a form of logical thinking that's going to use a general principle or law to forecast a specific result. So this is where we start getting into things like hypothesis testing. So past inductive reasoning conclusions. So, you're doing descriptive science, you're out in the world, you're discovering things, and you accumulate enough results to come to a conclusion. Maybe you even develop a law or a theory, right? And you use that, so let's say cell theory, you use that to then predict for a specific experiment. So a really interesting one is the example of what causes stomach ulcers. All right, well, you'll hear all sorts of things from people. Oh, it was from I ate too much acidic food, too much spicy food. But some researchers in Australia were like, well, we have people stop using, having food like that, and they still have stomach ulcers. What's going on? They did biopsies of a lot of people, people with ulcers and people who did not have ulcers, and they compared the tissue samples. And in every person who had an ulcer, they also had a bacterium called H. pylori, Helicobacter pylori. This little squiggly guy. Okay, is that enough to say for sure that H. pylori causes stomach ulcers? No. It's there when we see stomach ulcers. We can say that. And it's not there when they don't have stomach ulcers. Okay, so that's our descriptive science right there. So now we can form a hypothesis. If an individual with an H. pylori infection is given antibiotics, their stomach ulcer will go away. Oh, that's pretty good. Can we do that? We can do that, right? So we used inductive reasoning, descriptive science, to then get to a place where we could use deductive reasoning to make a prediction of an outcome. So we take some people with H. pylori infections and we give them antibiotics and we look and see what happens. Well, they did that, and lo and behold, those people, their stomach ulcers cleared up and they were fine. That still leaves a bit of a gap in the knowledge though. Like, it could have been something else. You know, it could be a coincidence. It's probably not, but still, you know, it's not always 100%. This is where things can get tricky. What would you need to do to be absolutely certain? Well, you would need to take a person who does not have a stomach ulcer, infect them with H. pylori, and see if a stomach ulcer develops. Uh-oh. Ethics. Is it ethical to do something like that? Absolutely not. No. Even if you ask people to volunteer to do it, even if you paid them to do it, that is unethical science. So, one of the researchers in the group who did not have a stomach ulcer infected himself with H. pylori, documented the entire process, and then had himself treated with antibiotics. And lo and behold, the H. pylori caused a stomach ulcer, and then the antibiotics cleared it up. So, yeah. But again, I don't recommend doing science that way, but that is a way that science has been done for hundreds of years, where the researcher sometimes has to take the risk themselves. All right, so let's see. Okay, so wrap that up. It can seem like hopefully my little story helped you see the clear connection between inductive and deductive reasoning, but a lot of the time people think, oh, you know, descriptive science and hypothesis-based, these are separate things. They're usually not. They feed off of each other as we're doing the work. Sorry, don't mind my dog. They feed off each other. We do some descriptive work, and then we use that descriptive work to develop hypotheses, and then we test the hypotheses, and then we make new observations, and on we go. All right. So applied versus basic science. Mentioned this a little bit earlier, but this is another good spot to kind of bring it back up. We use the inductive and deductive reasoning. We use descriptive and hypothesis-based work in both basic, pure science and applied science. All right. So, you know, it's what often happens is that people think, oh, well, you know, applied science, that has a really clear role in what's happening. Like, oh, we need to figure out how to cure this disease. Yes, applied science is super important. But sometimes it overshadows the basic, the pure curiosity-based science, where, you know, we're just doing it out of curiosity. Oh, this is odd. Why is it doing that? But so often, work that comes out of the pure space, it leads into the applied. You know, a really good example of that is the Human Genome Project. Oh, geez, most of you may not have even been born yet when it actually finished. But I was a kid when they started this project. I know it was a huge deal. And people were, it was all over the place. People were so excited because we could sequence genomes and we were going to look at human genomes from all over the world. And other people were mad because it was incredibly expensive. I think it ended up costing several $1 billion to get the work done. And it took 13 years. Researchers from a huge range of different fields, from biology and chemistry and physics, and engineers to create the machinery to do the work. I mean, it's just this huge, massive project. One of the problems that people had was, how is this going to help us? And they couldn't give a really clear answer. Well, we'll know what the whole genome looks like. Well, at the time, and you may have heard this term, People thought the majority of the human genome, and all genomes really, was made up of what they called junk DNA, remnants from viral infections and, weird duplications and such. And that junk DNA wasn't of value, that didn't do anything. Well, this project was finished in 2003, which I'll age myself. That's the year I graduated from high school. Since then, we've learned that stuff they were calling junk DNA, it's not junk at all. It's incredibly important and plays big roles in determining when genes are turned on and off. And we'll talk about that in a few chapters. But it turns out none of it was junk. It was all important. And if we hadn't done this, just pure curiosity, can we sequence the whole human genome for humans all over the world? If we hadn't done that, how much longer would it have taken to learn that all this stuff we were calling junk was actually very important? So anyway, it's important to remember that all the science is important to look at, not just the stuff with a clear, this is definitely going to help us do this one thing. Okay.